Photosynthesis is a crucial and also incredible process that plants use to convert sunlight, water, and air into energy and food. And it's not super efficient, but what if we could make it more efficient? And what if we could use that to help in our plant production? That's what one Cornell research scientist, Maureen Hansen, has said in her most recent research on the topic. And it could be a breakthrough.
It is a really interesting scientific breakthrough. I think he described it as that one of those eureka moments that you have when you work in science. And what it basically does is it took rubisco, which is the enzyme from these blue-green algae, and it combined it with two other bacterial proteins in plants, in the chloroplasts of tobacco, to be precise, is what this experiment was on. But it sped up the process of photosynthesis to up to 60%.
So it's producing those sugars much more quickly and much more efficiently.
Exactly, and rubisco accounts for over half of the protein in leaves, soluble protein. So it's something that could be used for food production, and it could do a lot of things. It could lessen the land that we need for agricultural crops. It could, the problem being, is that this enzyme is very prevalent in nature, but it's also not very efficient. That's why it's so common. That's why there's so much of it. And sometimes it wastes energy and it ends up reacting with oxygen instead of with CO2 to produce these sugars. So these plants need to be encased in a very CO2-rich environment, which is something that right now we don't really know how to control, and definitely these plants are not in.
So it's not really something we could easily replicate outside of a lab setting?
Probably not easily, no. But this could be applied especially to rice and wheat crops, which would be very interesting, and I think that's the intended goal for the next few years. And it could also reduce fertilizer needs. It's something that blue-green algae reproduces and photosynthesizes very quickly compared to the plants that we use for consumption. So they were very smart in doing it, and it's research that's been ongoing for a really long time. And now with the insertion of these other two proteins is kind of where they had their aha eureka moment.
But we're still a far way off from ever actually implementing this. Though it does seem like the research could be used to spurn other applications.
It does, and I think that a couple of tobacco plants, which again is the plant that they've been doing
Why was tobacco used?
You know what? I don't know why it was used. I mean,
perhaps it's a very well studied.
Maybe it's a very well controlled crop. Yeah. Your guess is as good as mine in why they chose tobacco in this case. Maybe the chloroplasts inside tobacco are special in some way, or probably the, I don't know, growth rate is very fast, the lifespan is fast.
I'd like to see it in wheat or rice, ideally.
That's what they're moving on to, hopefully, next with their studies. I think this is a very wonderful, kind of optimistic discovery, and something that we are now using GMOs to be able to fix some of those needs that overpopulation has caused, such as food scarcity and kind of malnourishment and things like that. We're now able to fix it.
There are several opponents to GMOs. I mean, when you say that word, it does evoke some emotion. Oh, not emotion, some response.
I think that GMOs are confused sometimes with some of the corporations.
Monsanto. We don't want to get behind Monsanto.
Yes, exactly. Some of their practices are very questionable and definitely easy to be opposed to. I am personally very opposed to some of those practices, but GMOs are different. What we're doing right now by modifying these plants and being able to increase food production could significantly improve the quality of life of some of these underprivileged, underdeveloped, malnourished countries.
And the research is very worthwhile in its own right. What do you think of speeding up the power of photosynthesis to help engineer our crops to feed more people? Let us know what you think below in the comments on this issue, and please be sure to subscribe.